There is another important player on the innate immune system team which can help defend against a viral infection, and that is the natural killer cell. Indeed, humans with genetic defects, which make them deficient in natural killer cell function, have great difficulty controlling herpes virus and human papillomavirus infections. Natural killer cells mature in the bone marrow, and when they are not responding to an infection, are short-lived with a half-life of only about a week. Most natural killer cells are found in the blood or in the spleen and liver. These are two organs that store blood, and relatively few natural killer cells reside in tissues that are not under attack. So, like neutrophils, natural killer cells are mostly on call. Natural killer cells use the roll, stop, exit strategy to leave the blood and enter tissues at sites of infection. And once in the tissues, they proliferate rapidly to build up their numbers. When they reach the battleground, natural killer cells can play two roles in defending us against infections. First, natural killer cells can give off cytokines such as interferon gamma that help with the defense. Like macrophages, natural killer cells can exist in several stages of readiness. Resting natural killer cells produce some cytokines and can kill. But they produce larger quantities of cytokines and can kill more efficiently if they are activated. Several signals have been identified that can activate natural killer cells, and each of these signals is generated only when the body is under attack. For example, during a viral attack, other immune cells produce interferon alpha and interferon beta that activate natural killer cells. Natural killer cells can destroy some tumor cells, virus-infected cells, bacteria, parasites, and fungi. They kill these cells by forcing them to commit suicide. In some cases, natural killer cells employ an injection system that uses perforin proteins to help deliver suicide enzymes such as granzyme B into a target cell. In other situations, a protein called Fas ligand on the natural killer cell surface interacts with a protein called Fas on the surface of its target, signaling the target cell to self-destruction. The method natural killer cells employ to identify their targets is quite different from that of killer T cells. Natural killer cells do not have T cell receptors. These are the receptors that are constructed by mixing and matching gene segments. The surface receptors that natural killer cells use for target recognition are of two types. One is activating receptors which, when engaged, motivate the natural killer cell to kill. Other is inhibitory receptors, which, when engaged, encourage it not to kill. The don't kill signal is conveyed by inhibitory receptors that recognize class 1 MHC molecules on the surface of a potential target cell. Class 1 MHC molecules are found in varying amounts on the surface of most healthy cells in our bodies. Consequently, the presence of this surface molecule is an indication that a cell is doing okay. In contrast, kill signals involve interactions between the activating receptors on the surface of a natural killer cell and unusual carbohydrates or proteins on the surface of a target cell. These peculiar surface molecules act as flags, which indicate that the target cell has been stressed, usually because it has been infected by a virus or is becoming cancerous. Natural killer cells measure the relative strengths of the kill and don't kill signals to evaluate the health of the cell and to determine whether that cell should be destroyed. Now, why do you think it would be a good idea to have natural cells destroy cells that do not express class 1 MHC molecules? You remember that by examining peptides displayed by Class I MHC proteins, killer T cells are able to look inside cells to see if anything is wrong. But what if some clever virus were to turn off expression of MHC molecules in the cells it infects? Wouldn't those virus-infected cells then be invisible to killer T cells? Indeed, they would be. So, in those cases, it would be great to have another weapon that could kill the virus-infected cells that don't display MHC molecules on their surface. And that's just what natural killer cells can do. Now, signal transduction events involved in natural killer cell-mediated apoptosis. Natural killer cells can kill target cells by two major pathways. In the cytotoxic granule-dependent pathway, binding of the natural killer receptors to the surface of the virally infected cell triggers the extracellular release of perforin, which is a pore-forming protein, and granzymes, which are a diverse collection of proteases from the natural killer cell cytotoxic granules. Let's understand both mechanism in detail. This is a natural killer cell. It has cytotoxic granules having perforin and granzymes. The granzyme and perforin are released from natural killer cells and target the cell. This is a target cell. Perforin bears some structural homology to C9. It is like that protein, but without any help other than from calcium. It can insert itself into the membrane of the target, apparently by binding to phosphorylcholine through its central amphipathic domain. It then polymerizes to form a transmembrane pore with an annular structure comparable to the complement membrane attack complex. So perforin polymerizes within the target cell membrane to form transmembrane channels that permit entry of granzymes into the target cell. Polarization of the granules towards the target cell takes place as a result of the formation of a synapse between the killer and target that is composed of an adhesion molecule called LFA1 on natural killer cell and its cognate receptor ICAM1 on target cell. Granzymes induce apoptotic cell death through activation of the caspase protease cascade, either by directly processing and activating caspases, or activate BID. BID action has been proposed to cause mitochondria to produce apoptogenic proteins like cytochrome C. So the release of cytochrome C from mitochondria occurs. It activates caspase 9. In the second pathway to cell death called the death receptor pathway, membrane-bound Fas ligand on the natural killer cell engages and telomerizes surface Fas receptors on the target cell. Engagement of Fas receptors recruits the adapter protein, Fas-associated death domain protein, followed by caspase 8, which then becomes activated at the receptor. Caspase 8 can then promote further caspase activation through directly processing other caspases, or through the mitochondrial apoptosome pathway, similar to granzymes. In both pathways, the final common pathway to apoptosis occurs as a result of the activation of several executioner caspases that coordinate cell death through restricted proteolysis of hundreds of cellular proteins. Next, we will discuss the activated natural killer cells can amplify immune responses through production of interferon gamma. Another consequence of the activation of natural killer cells is the production of another type of interferon, interferon gamma, a very important cytokine that has a set of activities distinct from that of interferon alpha and interferon beta. So type 1 interferons, or direct pathogen-associated molecular patterns mediated stimulation, activates natural killer cells. Let's discuss it. In macrophage activation by viral infection leads to production of type 1 interferon or interferon alpha and interferon beta that activate natural killer cell. Natural killer cell can be activated when pattern recognition receptors engages with pathogen-associated molecular pattern. These are pattern recognition receptors. These proteins are capable of recognizing molecules frequently found in pathogens, so-called pathogen-associated molecular patterns. Macrophages can produce type 1 interferons. Activated macrophage, leading to 100-fold enhancement of natural killer cell killing activity or activation of natural killer cells. Activated natural killer cells, leading to interferon gamma secretion, means activated natural killer cells are an important source of interferon gamma, which cause macrophage to lead the production of interleukin-12. Interleukin-12 is an important T-cell polarizing cytokine. This interferon gamma cause enhancement killing of intracellular microbes by macrophages, and that shape the nature of the ensuing immune response by T-cells within the adaptive immune system. Production of interferon gamma by natural killer cells also enhances antigen presentation by dendritic cells, which is also important for activation of the adaptive immune system. This cytokine can also influence the type of adaptive immune response that is mounted by helping to polarize T cells towards a particular response pattern. Interferon gamma also leads to enhanced target cell killing. This was all about natural killer cells. In our next section, we will talk about the example of innate and acquired responses working together to defeat the enemy.